Welcome to Chapters 21 and 22 review. Chapter 21 is about carbohydrate metabolism and essentially glycolysis, other pathways such as gluconeogenesis, glycogenolysis, and glycogenesis, the synthesis of glycogen. Also talks a little bit about digestion. Now, this document covers both chapters 21 and 22, but this video will only cover the first half, maybe the first 9 or 10 items here dealing with chapter 21, carbohydrate digestion. Chapter 22 deals with this common pathway that I discussed in class, namely the citric acid cycle. So, one of the main things to keep in mind when studying metabolism, do not get hung up on all the different intermediates and their structures. If you notice on this document, there are absolutely zero structures, unlike most of the other chapters that we've dealt with. So I'm going to be referring to these compounds by their names and not necessarily identifying their structures. The structures are kind of complicated and I wouldn't have you draw them, but you should have some idea of certainly what glucose looks like and certainly what ATP looks like. So this document doesn't go into nearly as much detail as we discussed in lecture, but it should be a good starting point. Covers a lot of the basic higher level concepts. So digestion now, it starts in the mouth. So here starts in the mouth. Okay, and as far as digestion of carbohydrates goes, it begins in the mouth with salivary amylase. And what we're talking about here is starches, essentially. So starch is amylose, amylopectin. Nothing occurs in the stomach and is completed in the small intestine. Okay. These starches in the mouth are converted to dextrins. These are essentially oligosaccharides, smaller. We haven't broken down starch into individual glucose molecules, but they're broken down essentially into smaller polysaccharides. These saccharides to give you a negative iodine test, as we discovered in lab, where I had you spit into test tubes. These eventually then in the small intestine, so I'll put here small intestine. These are converted to the monosaccharides, glucose, galactose, and fructose. Now naturally, glucose only comes from, is primarily from starch. Galactose comes from the disaccharide lactose, and fructose comes from the disaccharide sucrose. So I just want you to understand that when I'm talking about these dextrins here, I'm really only talking about the breaking down of starch, which only contains glucose. But the other disaccharides, lactose from milk and sucrose from table sugar, honey, other syrup, those are also broken down, not in the mouth, right, but in the small intestine. Glycolysis is a 10-step pathway that, and I should also mention that digestion, this is extracellular. It is enzymatic, so it is catalyzed by enzymes, but it is extracellular. Small intestine amylase, we have, we can read them right here. We can say here, this is salivary amylase in the mouth. Salivary amylase. Salivary amylase does not survive the stomach, but the pancreas secrete pancreatic amylase into the small intestine and complete this digestion, pancreatic amylase. Okay. Now glycolysis is intracellular. So the process of digestion, these monosaccharides are released. These go into the bloodstream. They're taken up by cells. And it's also not only in the cell, but it's also cytosolic. Occurs in the cytosolic. Okay. Essentially, again, keeping a broad, big picture of metabolism, you should know what goes into these pathways, into glycolysis, and what comes out, and then also how that pathway is regulated. So what goes into glycolysis? What is the starting material for glycolysis? Well, it's obviously glucose, but fructose and galactose also enter the pathway. The galactose is isomerized to glucose, and fructose enters, if you study the pathway, you'll see that glucose is isomerized into fructose. Glucose 6-phosphate converted to fructose 6-phosphate. And so fructose can enter in at that level. What else do we need for glycolysis? Well, we're going to need some ATP. Okay. So we're going to use 2 ATP, and we also use some NAD. And 2 NAD. That nicotinamide adenine dinucleotide coenzyme NAD. So that's what goes into glucose. What comes out, and I should mention, obviously glucose is a hexose, so 6 carbons. What comes out, right, what are the products? We have 2 pyruvates. Pyruvate is 3 carbons. I'm going to draw the structure just to impress upon you the fact that pyruvate does have 3 carbons. It's an alpha keto acid, 3 carbon acid, but we have a ketone here in carbon number 2. So we get 2 pyruvates. Now the ATP, actually I'm going to say 2 ATP and then also 4 ADP. 2 ATP are used in that energy investing stage, and that's occurs in the first 5 steps. The second half, I should say here, this is a 10-step pathway. 10-step pathway. The first 5 steps is an energy investing where we use up 2 ATP. The second half is where we're going to use the NAD and ADP, and essentially from this 4 ADP we are going to use and put here 4 inorganic phosphate. So PI, that little I means inorganic, so essentially it's just a phosphate ion. We refer to this as a substrate level phosphorylation, where this inorganic phosphate is added to ADP, so we get 4 ATP out. So what comes out is 2 pyruvates, 4 ATP. Now we used 2 ATP at the beginning to activate glucose, and then we get 4 out. So the net is just plus 2. It's plus 2 ATP. So for each glucose that goes in, we net 2 ATP molecules. And then also the 2 NAD is converted to 2 NADH. So 2 NADH. So a very quick summary of glycolysis. Conversion of glucose to pyruvate, and we get a little bit of energy. Understand how the energy charge of the cell is related to metabolic pathways. Energy charge, you can think of this as maybe the fuel gauge for a cell. It's signaled by high ATP. Put here high ATP equals high energy charge of the cell. One of the things we'll be discussing in the next chapter is the citric acid cycle, and it turns out high citrate levels also indicate a high energy level. So I can put here high citrate equals high energy, and as we'll also see, high glucose. This is cellular glucose, right? Because we're talking about the energy of the cell. So high concentrations of glucose also signal high energy. So The products of glycolysis is ATP, also NADH. So it turns out these reduced coenzymes also signal high energy. We'll see that maybe in the next chapter, but I'm going to say here high NADH, and also FADH2, although we did not see that in this chapter. FADH2 is also signals high energy. A low energy charge of the cell, well, it's going to be kind of the opposite. It's going to be signaled by, if you wish, a low ATP, or in other words, a high ADP for adenosine diphosphate and adenosine monophosphate. It's also signaled, this would be low citrate, so also signaled by low citrate or low glucose. But high, let's say high NAD or FAD. Right. NAD and FAD are the oxidized forms. They have not gained these hydrogens. Right. So high NADH and FADH2, the reduced coenzymes, indicate a high energy charge. The oxidized forms indicate a low energy charge. So when I'm talking about high here, I'm talking about concentrations. High concentrations of NAD and FAD, high concentrations of ADP and AMP. And then really what we're looking at is a ratio of ADP, AMP over ATP, or if you wish, the ratio of NAD over NADH. Catabolic processes are inhibited when the energy charge is high. You should know what catabolic processes are. Catabolic processes are the ones that produce energy. So if the energy charge of the cell is high, we don't need to make more ATP. We're going to inhibit these processes. Catalytic processes are the ones that break large compounds down, such as starches or proteins, into carbohydrates and amino acids, respectively. Conversely, anabolic processes are activated when the energy charge is high. So if we've got a lot of energy around, we can maybe store some of the glucose, right? High glucose and glucose, high energy. We can store some of that glucose as glycogen, converting glucose, a small molecule, into glycogen. We're building up a large molecule like glycogen. That's an anabolic process. All right, ATP is known as the energy currency of the cell. So it has a central role here. Most of what we're talking about in chapters 21 and 22 is essentially how do we get ATP? How is ATP formed from these carbohydrates or proteins or lipids? ATP, the T stands for triphosphate, but there are only two high energy bonds in ATP. We detailed that in lecture, the difference in the chemical nature. The high energy bonds are acid and hydride bonds. That third phosphate is linked to the ribose of adenosine. Adenosine is adenine plus ribose, so adenosine triphosphate has a ribose, and the third phosphate is actually in a phosphoester bond, not an acid and hydride bond. So there's a difference chemically in the nature of these bonds, and the two acid and hydride bonds are the high energy bonds, the two high energy bonds. All right, during catabolism, glucose is oxidized. Yeah, it's converted to carbon dioxide and water, but really, specifically, it is oxidized to carbon dioxide and water. You can do the same reaction simply by putting a flame to glucose and igniting it. Kind of get a really nasty mess. But when people determine the caloric content of foods, essentially that's what they do. They burn the food, they dry it out, so get all the water out, and then they burn it, and it is oxidized in that combustion reaction to carbon dioxide and water. Now, naturally, we do not have flames in our cells, so there is energy released by this oxidation, just as it is released when you burn something, but that energy is not in the presence of a flame. It's present in these the energy currency. Where did I write that down? ATP is the energy currency, and also in the reduced coenzymes NADH and FADH2. The oxidizing agents used in these reactions are these coenzymes NAD NAD, if you recall, is used to make carbon-oxygen bonds. So for example, we go from the alcohol to the carbonyl. Right, so we've made a carbon-oxygen bond. And FADH2 is the other, actually FAD is the other coenzyme. It is reduced to FADH2 in the process, but here FAD is used to make carbon-carbon bonds. So essentially carbon-carbon double bonds used to convert an alkane to an alkene. Right. During the process, as I mentioned, these coenzymes are reduced. These coenzymes NAD and FAD are reduced to NADH. Right. Reduction is a gain of hydrogen, so it's gained a hydrogen, or FADH2. Gain of hydrogen. Okay, understand how blood glucose levels are maintained and the role that insulin and glucagon play in this process. Key to understanding the action of insulin is to understand these two concepts. Insulin's function is to reduce blood sugar. In other words, reduce blood glucose levels, and this is by, it doesn't get rid of the glucose, it just it transfers the glucose from the blood, so it's extracellular, to intracellular glucose, glucose that's in the cells, glucose that is in the cells. So insulin reduces blood sugar by increasing cellular uptake. Glucagon, and I should also mention these guys are both peptide hormones. From the pancreas. Glucagon is really the opposite of insulin. Glucagon increases blood glucose. How does it do that? Well, one way you could increase blood glucose is simply by eating something that's high in glucose, something that's starchy or sweet and sugary. Okay? That's not what glucagon is doing. What glucagon does is it activates or mobilizes the glucose stored as glycogen. So it does decrease cellular uptake, but it also activates glycogenolysis. So glucagon increases blood glucose by decreasing cellular uptake and increasing glycogenolysis. This glycogenolysis, this is the hydrolysis of glycogen. So glycogen in the cells is broken down into glucose. That's glycogenolysis. And then the glucose in the cell is exported to glucose in the blood. Now it's mostly the kidneys. I should mention this is the kidneys and the liver that can do this. Muscle cells, other cells lack the enzyme to remove the phosphate on glucose. Glucose 6-phosphate is how glucose is normally kept in the cell, and it can't pass back out. And there is no enzyme in most cells to remove that phosphate, but in kidneys and liver there is a dephosphorylating enzyme. And so once you remove the phosphate off the glucose, it can travel out of the cell and into the blood and maintain that proper blood sugar level. Okay, what are the 4 fates of pyruvate? I think in lecture I talked about 5 fates, but the 4th fate is 4th and 5th are kind of the same. You'll see why in a moment. Alright, so first off, pyruvate is converted to acetyl-CoA, another coenzyme, coenzyme A. So I'm going to draw the structure here. Acetyl is 2 carbons. carbonyl there, and then coenzyme A. Actually, let me change this. It is in a thioester bond that I discussed in lecture, and this thioester bond is also a high energy bond, so I'll put a little line there indicating that this is also a high energy compound comparable to ATP. However, there's only one high energy bond here, whereas in ATP there's actually two. Alright, so it's converted, pyruvate is converted to acetyl-CoA, this compound here, when the citric acid cycle is active. The citric acid cycle is active. We'll see this in chapter 22, but it turns out the citric acid cycle is aerobic. It depends on oxygen. So when the citric acid cycle is active, essentially what that means is there's oxygen present, so plus O2. This means it's aerobic. Glycolysis is anaerobic. It's an anaerobic process, meaning it does not require oxygen. And that's what B and C are essentially discussing. So pyruvate is converted to lactate, say here in animals, when it's aerobic. Right, that means no oxygen. So in other words, no citric acid cycle. Let me draw the structure of lactate. Pyruvate, if you recall, was an alpha keto acid. This is an alpha hydroxy acid. So the ketone here on the second carbon is reduced to an alcohol, right? And then we get lactate. Pyruvate is converted to this lactate under anaerobic conditions. So when oxygen is absent, we talked about a number of different ways in which oxygen might be absent. There's strenuous exercise, certain drugs or disease states, or heart attack, things like that, that decrease the oxygen present available for cells. Now the reason animals do this is because in the process of converting pyruvate to this lactate, right, this is a reduction. I could even say pyruvate is reduced to lactate. What that means is something's got to get oxidized. In the process, NADH gets oxidized to NAD. And the reason the cell does this is because NAD, if you recall, is necessary for glycolysis to continue. We don't get a whole lot of ATP from glycolysis, only 2 ATP molecules per glucose, but we do get some. So this is a way for the cell to kind of limp along even in the absence of oxygen. So reduction of lactate, reduction of pyruvate to lactate results in the oxidation of NADH. Essentially, what this does is it recycles or regenerates NAD. And the key thing about NAD is this is necessary for glycolysis to continue. And I'll just put here as a reminder, we get 2 ATP out of glycolysis. And so why do we want glycolysis to continue in the absence of oxygen? So we can still get a little bit of energy from the glucose. Third fate here, pyruvate is converted to ethanol, or grain alcohol, or ethyl alcohol, which is a 2-carbon alcohol. So you notice we had 3 carbons. This COO here is converted to carbon dioxide, so it's converted to ethanol and also carbon dioxide. So carbon dioxide gas is formed. That is an oxidation. This acid group, this carboxylate group, getting converted to CO2 is an oxidation. That actually results in an aldehyde, and then the aldehyde is then reduced to the alcohol, and that's where again we produce NADH, or we recycle NADH to NAD. So also when oxygen is absent, so anaerobic conditions, but this is only in microorganisms or yeast. We'll just say yeast. So animals convert pyruvate to lactate. These microorganisms, yeast, convert pyruvate to ethanol and carbon dioxide, but for the very same reason, to recycle NADA so glycolysis can continue. Okay, the fourth fate, number 4, I'm going to put number 5 down here. Pyruvate is converted to oxaloacetate. Don't worry about what the structure of oxaloacetate is, just for right now. We'll see it in the next chapter, but for right now understand that it has got 4 carbons. Pyruvate is 3 carbons. So what we do is we're going to add carbon dioxide to it. So it is not just converted, but it is carboxylated to oxaloacetate. This is again when the citric acid cycle is active. Which means oxygen is present in order for the citric acid cycle to be working. And the reason for that, I'll just put here, citric acid cycle is also known as the Krebs cycle. So it's a series of 8 reactions, and the thing about it is what starts the citric acid cycle is a 4-carbon compound known as oxaloacetate. So in order for the citric acid cycle to work, you have to have oxaloacetate. Where does that come from? Well, it can come from some amino acids, but it also comes from pyruvate. And then we have our 2-carbon unit. This would be acetyl-CoA. And that gives you a 6-carbon citric acid. So 4 carbons plus 2 carbons gives you the 6-carbon citric acid, hence the citric acid cycle. The 5th fate is also oxaloacetate, but for a different reason. This is when glycogen or glucose is low. We need to make new glucose. So this is when gluconeogenesis is active. Essentially what we're going to do is take pyruvate and convert it back up to glucose. Well, actually the first step in that process of gluconeogenesis is to carboxylate pyruvate, C3, add another carbon to get oxaloacetate. Glycolysis. It's activated when energy charge of the cell is low. And that's signaled by what? That's signaled by AMP. Also AD, I should put here ADP, AMP, and also insulin. Insulin activates glycolysis. It's inhibited when the energy charge is high. Okay, well, why would it be high? Well, we've got a lot of ATP, So it's inhibited by ATP. One of the products of, not directly, but one step removed from glycolysis is that compound acetyl-CoA. And so this is a type of feedback inhibition that we discussed, where the product here, acetyl-CoA, inhibits its own synthesis. So where does acetyl-CoA come from? Well, pyruvate, which again is 3 carbons, instead of adding a 4th one, we remove that carbon to get acetyl-CoA. That enters into the citric acid cycle. So this acetyl-CoA can sort of be considered a product of, not directly, but a product of glycolysis. And so we're going to slow that reaction, that series of reactions down when we've had a lot of acetyl-CoA and a feedback, allosteric feedback inhibition. It's also inhibited by high levels of glucose 6-phosphate. I just put here glucose 6P, glucose 6-phosphate. If you remember, glucose 6-phosphate is the first thing that happens to glucose. Hexokinase phosphorylates glucose 6-phosphate, kind of traps it in the cell. But glucose 6-phosphate has 2 fates. It can either go through glycolysis, or it can also be, it's also the first step in glycogen synthesis, or glycogenesis. So if we've got a lot of glucose 6-phosphate around, maybe we have enough ATP, we don't need to make more, so we're going to store this glucose as glycogen. So when a lot of glucose 6-phosphate is around, we're going to slow down glycolysis and actually speed up glycogenesis. Gluconeogenesis, this is the creation of new glucose, so making glucose anew. I mentioned that up here. First step is to add a carbon to pyruvate to get oxaloacetate. This is the first step in gluconeogenesis. So when do we need to make new glucose? Well, it's going to be activated by, obviously, low glucose. And glucose's concentrations are low. Gluconeogenesis speeds up. Remember, insulin and glucagon are kind of opposites, whereas glycolysis is going to be activated by insulin. Gluconeogenesis, which is making new glucose, not tearing it down. Glycolysis is breaking it down. Gluconeogenesis is making new glucose, so it's going to be activated by glucagon. Also notice, if glycolysis is inhibited by glucose 6-phosphate, as I mentioned before, it's going to be gluconeogenesis is going to be activated by glucose 6-phosphate. So we see this kind of diagonal relationship. So why or when would gluconeogenesis be inhibited? Well, it's going to be inhibited by, so we'll have this diagonal relationship here as well. AMP is going to inhibit gluconeogenesis, as does insulin. Gluconeogenesis is inhibited by AMP and insulin. And if you think about it, what's the product of gluconeogenesis but glucose? Right? So if we've got a lot of glucose around, why do we need to make more? We don't want to make more, so we're going to inhibit this gluconeogenesis when we have high levels of glucose. Glycogenesis. Glycogenesis is making glycogen. So this is, you can think of this as glycogen synthesis. All these words sound alike. You kind of have to keep them straight. Gluconeogenesis is glucose, not glycogen, but glucose, the monosaccharide glucose synthesis. So when would we want to make glycogen? Well, if we have a lot of glucose around, more than we need for immediate use, we can store it as glycogen. What signals the cell that there's a lot of glucose around? Well, that's insulin. Right? So glycogenesis is going to be activated by insulin. What do you suppose it would be inhibited by? Well, it's going to be inhibited by its counterpart here, glucagon. And also that other hormone, epinephrine, the fight or flight hormone. epinephrine or adrenaline. Glycogenolysis. Glycogenolysis, this is the breaking down of glucose. Breaking down of glycogen. So it's the hydrolysis, breaking down of glycogen. And when do we need to do this? We need to do this when our blood glucose is low. So when blood glucose is low, what does that mean? Well, high blood glucose is signaled by insulin. Low blood glucose is signaled by glucagon. So this is going to be activated by glucagon and inhibited by insulin. So if you think about it, we've got glycogen, storage form of glucose. This can be converted down to glucose. That's glycogenolysis. And we can take glucose back up to glycogen. That's glycogenesis. Glycogenosis. Glycogenolysis. And I'm going to put a plus sign here. Glycogenesis, right? It's activated by insulin, so I'll put plus insulin. Insulin, if you want, you can put up arrow. Insulin is going to be inhibited by glucagon. So inhibited by glucagon, down arrow. And on the other side here, it'll be just the opposite. Right? On the other side here, insulin is going to decrease glycogenolysis. Where can I put that? Let's see. Insulin decreases glycogenolysis, but glucagon increases glycogenolysis. So it's important to understand, keep all these terms straight, and understand what activates them, what inhibits them, under what cellular conditions of glucose or energy are these activated or inhibited. Okay, number 10 here, this is going to be found in chapter 21. No, sorry, chapter 22. We're in chapter 21 now. So in chapter 22, we'll cover the rest of this document.